Hello, this is Dr. Rajiv. Today we will talk about regulation of cell cycle. In my previous video, we had talked about basic of cell cycle, its phases, and events in those particular phases. And since I had promised that in my next video I will present the regulation of cell cycle and the particular drug actions in those events of cell cycle. So now I came with regulation of cell cycle in this video, and before starting with the regulation of cell cycle, I will give you some events. I will give some points regarding the events of G1 and G2 phase of the cell cycle. I had mentioned in previous video that in G1 there is repair of the defect of nitrogen bases. Defect. If there is damage of nitrogen bases, then it is corrected in G1 phase, and if there is any defect in RNA replication, error in DNA replication, that is corrected in G2 phase. So this two, G1 and G2 phase are usually, if these defects are there, then they are corrected in the checkpoint between G1 to S and G2 to M. So there are two checkpoints, as I mentioned, G1 to S, in between G1 to S and G2 to M, and these checkpoints, they screen the defect in G1 and G2. If there is a defect like nucleotide base damage, or in G2 DNA replication error, so these are checked in these two checkpoints, and that is done particularly by two important proteins, and those are cyclins and cyclin dependent kinase. So how they do it, I will talk in further of this video. And what they do is, if the DNA of the cell is normal, then they allow the cell cycle to progress from G1 to S and then G2 to M. But if there is damage of DNA in the cell, then what they do, they correct it first, they repair it first, then they allow the progress of the cell cycle. But if the DNA damage is so severe that they are not able to correct that DNA damage, then the cell will go apoptosis. It won't be repaired. Okay. Now I will discuss about how cyclin and cyclin dependent kinase works. What happens that cyclin dependent kinase is a protein that is produced by G1 phase cell, and they remain inactive during its production, and this cyclin dependent kinase is activated by cyclin. Okay, so after activation of cyclin dependent kinase, what it does, it phosphorylates RB protein, RB protein, it phosphorylates RB protein, and after phosphorylation of RB protein become inactive. Okay, so inactivation of RB protein allows the cell to progress from G1 to S phase. This normally happens, but it may create problem, or it may be harmful to the cell if there is any mutation in the cell. If there is any mutation, or if there is any defect in DNA, then that may be harmful for the cell, because without repair of this DNA damage, it progresses from G1 to S phase, isn't it? And that defect replicates and go on. The damaged DNA will be circulated in cell cycle, and it will be very harmful. So what it does to check out that, to correct the DNA defect, P53, there is another gene called P53. This P53 inactivates cyclin dependent kinase. if there is any mutation or any defect. So P53 inactivates cyclin-dependent kinase and allows cell enough time to repair those defects if there is mutation or if there is any DNA damage. So the major role of P53 is if there is any defect, if there is any mutation that is harmful for the cell, then P53 inactivates cyclin-dependent kinase and allows cell enough time to repair. By those I have previously mentioned in previous video, that what DNA polymerase, endonuclease and ligase. Endonuclease just break it, and polymerase and ligase just fix it, the defect. And as now after the DNA defect is repaired, then cell will progress in its normal cycle. Thus, due to this major function of P53, it is also called the master of the cell. I think this is enough to know about regulation of cell cycle, and the specific drug in events, different events of the cell cycle, I will talk in another video. Thank you.